Amino acids are biomolecule having an amino group and a carboxylic acid group, and they are attached to a alpha carbon. Okay. It is said that there are more than 300 amino acids are present in our nature, but only 20 different amino acids are commonly found in protein. In addition to these 20 amino acids, there are many less common ones. Some are the residues modified after the protein has been synthesized. Others are amino acids present in living organism, but are not as a constituent of protein. Now, these the common amino acids of protein have been assigned 3-letter abbreviations and a 1-letter symbol. Okay, the name derived from the source from which they are first isolated. For example, in the case of asparagine, actually it was first isolated in asparagus, then glutamate isolated from wheat gluten, then tyrosine from cheese, and glycine from sweet. This cheese Greek word is tyros. From that word, this tyrosine, the name tyrosine is derived from a Greek word tyros. Like that, sweet in Greek it is glycose. Okay, so from that word glycose, the name glycine is derived. Okay, so our amino acid, as I said earlier, it is a biomolecule having an amino group. This is our amino group and a carboxylic acid group. Here it is. They are attached to a carbon atom, and this particular carbon atom is called what? Alpha carbon. Okay. In addition to this, we can see there is a hydrogen atom. There is a hydrogen atom and a R group attached to alpha carbon. Okay. Now, the carbon atoms are numbered from one end, giving the highest priority to whom? Highest priority to C1. This is our C1. Okay, to the carbon with the highest atomic number. So we are giving this as number 1 because this particular has got a highest atomic number. Okay, so the carboxyl carbon would be the C1 and alpha carbon would be the C2. This is our alpha carbon. C2 is our alpha carbon. The additional carbon atoms present in the R group. Okay, these are the additional carbon atoms present in the R group. Okay, and we are giving names like what? Beta, gamma, delta, and epsilon, like that. So here we are having a second carbon that is an alpha carbon. So our amino group, coming to this picture, our amino group is attached to which carbon? The second carbon, that is called alpha carbon. So we call them as alpha amino acids. Clear? I hope it is clear now. So now we got alpha amino acid, and here we are having R group. This is our R group. So this R group is specific to each amino acid. So every amino acid has have its own unique structure and characteristic due to the presence of this R group. Sweeter ion. See, at physiological pH, amino acid exit like this. This is called sweeter ion. Here, our amino group is protonated and carboxylic group is deprotonated. Chiral carbon. See, all amino acid except glycine. This is the structure of glycine. Here, the alpha carbon is bonded to 4 different groups. What are they? They are, we can see a carboxylic group and an amino group, an R group, and a hydrogen atom. Okay, so here our carbon is bonded by these 4 different groups. And so the alpha carbon in this atom is a chiral center. It is a chiral center. Now take the case of glycine. Glycine, we can see an additional hydrogen atom in the place of R group. So this is the structure of glycine. It is an exemption. So it is called a chiral carbon. So this chiral center, the presence of this chiral center, which actually results in the formation of 2 possible stereoisomers for this amino acid. As I said earlier, the carbon act here is our alpha carbon, this is our alpha carbon. Carbon act as chiral center and exhibit 2 different spatial configurations which are optically active and mirror images of each other. They are called enantiomers. Clear? The 2 amino acids are L amino acid and D amino acid. In L amino acid means levorotatory and D is the dextrorotatory. And for the L amino acid, the amino group is present on the left side, and for the D amino acid, it is present on the right side. And our amino acid residues in the protein molecule are exclusively L stereoisomers. Very, very important. Okay. So in the case of D amino acid residues, actually they have been found only in a few genetically small peptides, including some peptides of bacterial cell wall. Okay. All other amino acids are L amino acid. See, take the first case. Here we are having a amino acid, and we can see that our amino group is free. Okay, so it can form bond with any other amino acid. Here it is free. Suppose this amino group is not free and that is bonded to a bonded in a ring-like structure. That type of amino acid is called what? Amino acid. This is the example of amino acid. This is the structure of what? Proline. And here we can see that our amide nitrogen is bonded to alpha carbon and carbon from the side chain. So it is an amino group. Okay, these are the list of essential and non-essential amino acids. Essential amino acids are not made by human body and must instead be acquired from our diet. In the case of non-essential amino acid, they are synthesized by human body, and it is around 9 are found to be essential, and there are 11 amino acids which are known as non-essential amino acids.